let's get started. So first of all, thanks everybody for joining. And I know it's extremely last minute because basically we decided on, say, I don't know if it was Wednesday or Thursday, that it was a good idea to start doing more regular updates on the work that we do, especially the research side of carbon copies. And so we sent out this invite and quickly put together a presentation. So yeah, if anything seems a little bit rushed or hasty, you know why. We are planning to keep doing these fairly regularly. The next one may in fact be about 2 weeks from now, and we hope to then show you a more end-to-end run of the process as well. So you'll get more information about that eventually. An important thing for today's session is that we announced it as sort of a micro workshop and not as a talk. And because of that, it would be wonderful if you guys can, not hold back. Please use the little raise your hand icon, that little button in the Meet session if you want to jump in. It is okay probably to just start talking if there's already a quiet moment, but hand raising is typically slightly better, especially if there are multiple people who want to say something. And then what will happen is that, if we're on a slide and I'm discussing something and then there's a question or a bit of discussion that makes a lot of sense to talk about at that moment, then of course we will. If it's something that I'll be addressing in a slide later on, I might just say something like, oh, hang on, what you're talking about is actually coming up in a few slides. If you can just hold on and then we can talk about it there. But yeah, so hopefully this will be a nice sort of conversational setting. We're not in a gigantic rush, but we think it'll take somewhere between 60 and 90 minutes to get this thing done. To give you a little bit of a preview of the topics that we're going to go through, just in case any of you have to leave early or need to decide at which point you want to be absolutely present, we are going to talk about this thing called the Brain Emulation Challenge that we're working on, and we're going to talk about the reasons why and what it is. And we'll also talk about how the process works, so the thing that we've implemented, the technology we've created, parts of that anyway. And then we'll talk about how this addresses certain problems in the field of brain emulation, especially the questions of what we're calling translation and scale separation, which maybe some of you know from complex systems theory and other related domains. And then we'll talk a bit about what we hope that this brain emulation challenge will be like in the time coming up. So sort of the plan looking forward, and then our needs. So things that we need to make this happen properly, things that we hope that some of you guys or someone else will help us make possible. Okay, and the biggest part of this is going to probably be the part where we just demonstrate the technology that we've been working on and how it works, how the process works. With that, I'll jump right in. And can we go to the next slide, please, Thomas? Oh yeah, I should mention that I'm going to be saying that a lot because Thomas is helping me run through the slides so that I don't have to look at that. Instead, I can see if you guys are raising your hand, and we can do it that way. All right, so the first part is the question, what is the Brain Emulation Challenge? Why do we do the Brain Emulation Challenge? And when I say do we do the Brain Emulation Challenge, why are we working on a platform to support that, launching a challenge? What is it even? Let's go on to the next slide. All right, so the reason why we're interested in this thing called the Brain Emulation Challenge, why this is important to me, the work that we're doing right now, is because what I normally do and what we always do is we look at the landscape of everything that's going on in the field, in the various fields that are around emulation, about brain emulation, about that problem. And there have been very significant strides, big strides in data collection over the past few years. So, for example, the whole fruit fly brain has been scanned, at least with electron microscopy. And what happens is you end up with these very large piles of data, if I may call it that. They are high fidelity data sets. And we see that there are going to be many more of those data sets coming up. And now, as people are getting into the whole mouse brain project, Along comes another technology. There's this new expansion microscopy with molecular annotation and all of that that looks like it might displace electron microscopy, at least in some areas. So there's about to be another step change in data collection. So data collection seems to be doing well, even if we can always want more, of course. But despite all of this very detailed data collection, post-processing of the data, annotations being made now by machine learning as well as humans that are doing proofreading and identifying cell types in the connectome, ion channels, all sorts of things like that. All this identification going on, a 3D reconstruction being built with a lot of identification in it. There are really still very few examples of any sort of functional reconstruction. And I'd say there's really no example of a fully developed and validated functional reconstruction of, say, a tiny animal, even a fruit fly or a C. elegans or something like that, or any significant piece of neural tissue like that. I hope that I won't have to say that much longer. I hope that we'll soon run out of this constant refrain of, but still, there's no examples of that. But right now, this is still a problem that we have. Can we go to the next slide? So I think it would make a gigantic difference if we had a proof of concept of this whole idea of brain emulation. I think it would make a difference. Maybe it wouldn't solve the underlying philosophical questions right away, like really fundamental things about whether functionalism is true or something like that. But it could certainly help us understand how far you can get in terms of reconstructing a neural circuit and getting it to produce the same results and to capture those things that you actually care about. This capturing what you care about is really a very important aspect that I'll get into more as we get into how the brain emulation challenge works and why we chose to do it a certain way. But yeah, so there is that desire to see if it's even possible and what it takes, at which resolution you have to work, what components you need to identify, and what parameters you need to identify to make models of those components. Now, the big thing standing between the big pile of data, this high fidelity data that's collected, and having those functional models is something that, well, I know for sure that Ken Hayworth and I have started calling it the translation problem. I'm not sure if that term is widely known, or if it's just something we're introducing. But I like that term because it really is about the translation of measured data into working systems. And doing that in a specific way with a certain sets of success criteria and metrics in mind. I think it's actually surprising when you look at the big pile of data that's there, that there aren't already more people working hard on that problem. And there are, of course, some, and I'll just mention Conrad Cording as one who clearly sort of honed in on that and decided, yes, this is the next thing we need to do. I want to run my lab on that, and I want to use C. elegans as my ground truth system to work on it. There are others, of course. For example, there has also been a nice attempt by Philip Hsu to functionalize the fruit fly brain, the connectome, even though that connectome is mostly quiet, but it's some of the neurons get functionalized, and then you can see downstream propagation of activity. That's interesting. Still not quite the same thing. There's still a difference here between where we should be and where we are in terms of even just the attention in academia and outside academia to the problem. I have, at some other talks, I've sometimes gone out on a limb and I've said things like, oh, I think that, if we really understood what we were doing, if we knew how to do this translation properly, that probably the amount of data currently collected in the fruit fly brain, data stack, that is sufficient together with what we know about functional parameters of, say, models of neurons in fruit fly, that there is enough there that we could probably reconstruct that fruit fly brain and have an emulation of it. So in the future, the data that we have today would potentially be enough to do it. And maybe that's going out on a limb a bit far. That may not be 100% true, but I give it a fair odds that, if you really knew what you were doing, If you understood your blueprints and everything, that probably would be possible. So the question is, why? What do we do about it? How do we get more people into this? I mean, and I don't want to blame anyone for continuing to work on, say, data collection. It's important. It's not entirely like bike shedding, where an engineer will choose a simpler problem to keep working on that instead of the problem they should be working on. What color do you paint the bike shed instead of solving the problem? It's not quite like that. It's still important to improve data collection, but I really would like to see more work going on this translation problem. So let's go to the next slide here. Let's compare this with what we see in the world of artificial intelligence and machine learning. What do they do to produce models that use data, large data stacks that you can find in the world? Maybe data stack isn't the right word, but huge amounts of data in the world, and use it for something like, for example, autonomous driving or something else. What do? Well, you try to train up your model to understand something about the world based on that data, using data that we understand really well, fully understood ground truth data in a sense. The examples here, just showing you a couple of bicycles. It's just, sort of, typical type of thing you would find in a computer vision stack of data that might be used to train up a system to understand what a bicycle looks like. Looking at the bicycle in lots of different ways. There are sketches of bicycles, there are deformed bicycles, bicycles in snow, occluded bicycles, and so forth. You kind of teach it what that might be, and then eventually you can let the algorithm try its luck out in the real world, really identifying bicycles as your Tesla is driving along. So this is the sort of thing, not the only thing, but it's the sort of thing that happens in AI and machine learning. Now, the really useful thing they've got here is that they have all of this annotated data where they know there's a bicycle here. And so you can, and maybe there's a tree in the same picture as well. So you can give your system, as it's learning, feedback telling it, yep, you were correct. There is also a tree here and there is a bicycle, but no, there isn't a car or something like that. And all of that helps develop this process that you're trying to build up, the AI in that case. So the question is, how do you do something like that for neuroscience? And for that, let's go to the next slide. So here I'm just calling this the improvement loop. It's the idea is to have something similar to what's going on in AI and machine learning, where maybe it's not built into a computer, it's not just a machine that's doing this. These are neuroscience labs, researchers, the whole community is trying to do this loop where, down at the bottom, you've got that square collecting data, and then you want to analyze and interpret that data. You build a three-dimensional reconstruction, perhaps, because this was EM data. So this is data from which you can create a three-dimensional static reconstruction. Then there's that translation step where you attempt to build a dynamic model, you parameterize it. And then the next step, of course, because this is based on the same example as what we see in AI, and that really is the scientific method, actually, is that you try to verify your hypothesis, your model, right? You try to test it. So you want to verify and validate what you've done. You can't just functionalize the fruit fly brain. With all respect to Philip Shue, you can't just functionalize it and then claim, oh, I built the fruit fly brain, because how do you know? How do you test? How do you validate that this thing is correct? So you want to have a way of getting errors back, getting some corrections that you can make. You need to have real metrics. And then you feed that back into figuring out how to improve your methods. Or instead of improving your methods, maybe you're competing with someone else. You might be competing with another lab or another algorithm, something that was published somewhere else. This improvement loop can be quite big, but it's the same basic principle as what we saw in AI here. And the idea that there should be an iterative loop like this, that there should be an improvement loop, this isn't new, of course. It's not something we invented or anything like that. But it does take a moment to really realize that this is what you should be doing if you're trying to work on whole brain emulation. or any kind of emulation or neuroprosthetic or very detailed reconstructions of neural circuitry, you have to realize that this is in fact where we need to go to make it a robust, rigorous, concrete process, something where you're not just having one, say, moonshot idea, throwing everything at the wall, making some claims, but there's no way to test how well your method is doing and to compare it with another, unless somebody happens by accident to create a perfect emulation and it just works. But as modelers, we know that that's never the case. The first time you press run, nothing happens. The system's just sitting there dead. The fruit fly's not moving. Or it's got an epileptic seizure and you don't know why, and you don't know where the error is, right? So in modeling, you don't usually get that lucky. I mean, maybe there are some crazy lucky modelers out there, but I've not seen it yet. So you need to build a loop like that. And that was kind of our first realization back in 2023 when we went to the workshop that was held in Oxford that year about whole brain emulation. And we presented this idea of a process or a project where we would iteratively try to test all kinds of methods and keep on improving them. We had a name for that back then. We called it Next Up. Now, since then, we honed this idea quite a bit more, and we started focusing on a much smaller part of it. And that is that we really needed a platform first, a method where we could get not just us, but everybody in the world, academia, non-academics, more interested in working on the problem because there's a better way to work on it, a better way to actually compare methods, to contrast them, to get these metrics and these errors and be able to say this one has a much better performance score than that one. So we needed a place where people could actually practice doing translation. There's really this need to not just be able to grab some data that you collected from a piece of tissue where you don't really know what's supposed to be going on in there and then try to build a model from it, but to have really laboratory conditions where you can introduce variables one at a time, carefully measure performance at every difficulty level, and just ramp up the problem as you go. Can we have the next slide, please? So that's really the process of the brain emulation challenge that we then came up with. So instead of us trying to do this iterative loop ourselves right away, we decided that the right thing to do was to try to provide something for ourselves and for the community out there and for everybody really that would help sort of help get that circle going, that cycle going, and help increase the number of people interested in the problem. So what we're doing here really is we're trying to provide the kinds of data on which you can practice this, the same way as in AI, where you have, say, ImageNet and other data sets that are used to practice and train and improve methods on, where you know what you're putting in. You've got this ground truth. You understand that there are bicycles and cars and trees, and you can even do that in more detail. You can weigh how much does this look like a cat or a dog, perhaps, and things like that. So that type of data, fully known ground truth, not just ground truth, is what we need. And I'd like to make that distinction here, because when I talk to my neuroscience colleagues, for example, in microscopy or in more, say, experimental neuroscience and in other areas, is of course ground truth is the animal or the human. Ground truth is the biological system. It's the neural tissue. But it's not fully known ground truth. It's not the system where you can say, oh, here's the error I made when I reconstructed this. You just can't tell. So for this training ground, we need a fully known ground truth. And that is the first thing that this platform needs to be able to produce. It needs to be able to deliver that. And we weighed a few options. We weighed the option of going in with software, doing it virtually. We weighed the option of using neurons in culture, where you can pretty much see what's going on and record from everything. That second step, culture, is still something we really would like to do. Eventually, this is a plan. But right now, the more flexible thing is the virtual one. So that's where we start. And it's what you would do anyway, because even if you wanted to work on the biological method, or even if you wanted to reconstruct actual tissue from fruit fly, there's no doubt that anyone who's a modeler working on that, who is building up their methods, would eventually be doing something like generating some virtual data to test their methods, to test their programs, to test all of that. That's going to happen anyway. That's something you have to do. So why not just start there and build something really flexible? So this is what we do. If you look at that box bottom left here, embedded ground truth system, that's an abstract representation. You'll see more details in just a bit. But this is the idea. There is you've got a system where we choose some sort of function. And when I say choose, it can be a random pick from a bucket of possible functions. These are functions that have some, let's call it cognitive meaning. So for example, it could be auto-associative memory. It could be combining simple cells to complex cells in some sort of visual system stack, or something like what CA3 and CA1 are doing in the hippocampus. Whatever it is, the idea is that there's a meaningful function that we know that is there, an IO function. Why are we looking at that? We're looking at that because if you think about the criteria we most care about for brain emulation, we care most about those things that we use to create experiences, those things that animals use to live, behave, and experience, or that humans use to live, behave, and experience. Meaning, we don't care so much about which neurotransmitters being used. I really honestly can't tell what neurotransmitters are being used in my brain right now. But we do care about this functional IO, this functional processing of data as sensory information is coming in, as my thoughts are happening, as I'm turning that into some kind of behavior or decisions. Free will is a whole other question, but decisions are being made, whether they are free or not. So in any case, these functions are really what you care about if you're trying to create an emulation. If you ask somebody, what would you like to preserve if you're going to have whole brain emulation carried out? Well, they'll say things like memory patterns, which of course only mean something when they're being interpreted by some downstream system that can retrieve the memory, but they also need to be there. Or, the ability to see and all these functions, right? So we want to be able to make sure that the methods that are being developed in translation, that whatever they do, whatever type of data is being collected in the brain, and however you're treating that, however you're analyzing it, that you actually do discover those functions that are in the system that you're trying to get out. Now, because we're making a fully known ground truth system, we want something where we can tell whether the functions that we wanted to find have in fact been found, meaning we're putting something in on purpose. We're generating some function, and that is what's going in. Now, to make it more realistic and more difficult for these methods being developed, we of course want to make it realistic. We want to get more into the realm of what does that data normally look like. So we need a platform that can make something that looks like neural tissue. And I'll get into that a bit more in another slide, but we're using a platform that I worked on some years back called NetMorph that has models and methods and a stochastic approach of how various types of neurons grow. And we use that to generate, let's say, a block of virtual tissue where the neurites create connections that enable you to have a connectome that can support the function that you've put in. And after some tuning and pruning, then that gives you something that has somewhat the appearance of realistic neural tissue, or at least the neural circuit that's there. There's some more work that goes into making it more like neural tissue, and that can carry out the function that we were putting in. So we now have a something that is kind of virtual tissue with an embedded or hidden cognitive function in it. So, yeah, let me just see where I was here. Oh yeah, right. Okay, so the next box, I can't believe it, but this was all about the first box in the bottom. What happens then, of course, is that our system has to, or this platform, the thing that we're trying to create here, has to generate these data stacks that are like the data stacks that you would acquire or similar in some manner, maybe simplified and then made more complex as you go, like the data stacks that you use in neuroscience when you're really doing microscopy, when you're really doing electrode recording, when you're really doing calcium imaging, and so forth. And then you give that to the challenge participant, someone who's taking this challenge, and the participant, the challenge taker, will take that data, apply the methods that they have, for example, 3D reconstruction and then some sort of translation, to produce what they think is the emulated system in there. And that's what comes back to the challenge and gets evaluated. So there's a comparison where we see, okay, how much of the structure did they find? How much of the function did they find? I'll get into a little more detail about that later, but just to give you an idea of what this loop is here. So this whole thing, it's meant to make it easier for people in academia as well as outside of academia to get involved with this problem and to really deal with translation. And hopefully because of this feedback loop, then, there are going to be opportunities for us to tell them the sorts of errors that they're making and how to fix their methods, or at least how to compare one method with another. So you can see how this is a bit like the AI version, the AI kind of approach, but then for neuroscience. All right, let's go on to the next slide, which I believe opens up our next section or next subtopic. I'm going to take a quick sip of coffee before we get into that. Also, I haven't seen any hands raised yet, and I hope it's just because everything I'm saying is so obvious that there aren't any questions. But if there are, please feel free to ask something now. Or otherwise I'll just continue. Okay, so no pressing questions yet. Okay, let's go to the next slide. Oh, wait, 1 second. Oh no, that's just Alex saying no questions so far. All right. Yeah, let's go to the next slide. Okay, so how does this work? What does our system actually do? So the essential part, as I mentioned, of the Brain Emulation Challenge is the software platform that allows us to generate this virtual tissue and the acquired brain data from that. And the very first step, as I also alluded to, is to pick from a bucket one of a number of prepared types of cognitively relevant functional operations, which of course can be generated rather than being a fixed static thing. And then you can pick either one example or a combination of those examples. So for example, here, we've got on the right there, one example is auto-associative memory, because, maybe we wanted to test discovering something that contains circuitry that could enable auto-associative memory. So in that case, the system will require things like pyramidal neurons with recurrent connections, and it might also require inhibitory neurons that are recurrently connected with those because you're trying to separate patterns that you're encoding or retrieving or something like that. So this sets up a certain set of requirements if you pick that function. Or we could have chosen a propositional function like a spiking XOR operation, something that's a feed-forward neural circuit that just happens to be in that piece of tissue, carrying out something like XOR. And then, for instance, you can express that in terms that can be done with a neural operation. Like you can say it's A and not B, or B and not A, which means that you then have to use excitatory connections for things like your ands and your ors, and inhibitory connections for the not. It is a somewhat unusual example because it's not the sort of thing that a neuroscientist would usually say is, oh, this is what the memory does. Here's an XOR circuit. That's not what you normally come across. But propositional logic is something we do, even if we do it differently. But the most important reason why this example is in here is because it's super minimal. It's really tiny. That makes it an easy one for us to run through the next parts of this because it'll be easier to show what's going on just because it's smaller. Okay, so let's go to the next slide. All right, so after we've picked the function, or the system has picked the function that we want to embed in there and hide in there, the next step is to try to grow these realistic connectome reservoirs. And I assume it's running down there. I can't tell if that, oh, it is, yes. So in the corner down there, you can actually see NetMorph doing its business. It's a bit of a slow animation, but yeah, it just basically does a simulation from, say, 0 days to 21 days or longer if you want to. We typically run them for 21 days because that's about the number of days you need, say, in mouse or rat to establish connections that will produce things like theta oscillations and gamma oscillations. This is all based on work that I did for 4 years with Professor Jaap van Pelt and Professor Arjen van Ooyen in Amsterdam. NetMorph is a platform that uses both phenomenological and stochastic hypotheses that are built into the system to generate what seem like the type of structures that you would find in typical neurons, like a pyramidal neuron that would have like an apical tuft and obliques and all that sort of thing, and then give you automatically intrinsic things like if the branches always happen at a certain distance, then you typically get connections that will produce, say, rhythms of a certain kind. So there are a lot of things already built into what this platform can do to generate neural circuitry. We use it here in a very extended form. We've added a bunch of, say, controls, features, tweaks, variables, and an API to it so that we could embed it in our bigger platform that I'll talk about in a bit. And so that we could use it to grow out what is, let's say, a reservoir of possible connections that we need for these functions that we decided to put in there. And you could say that this first stage resembles the proliferation phase of human development, where you connect more things than you need, produce more synapses than you need, maybe have more neurons than you need to generate these reservoirs of potential that you can use. And I think I even got ahead of myself a little bit, because I should have said that determining that we needed to do this in 2 phases like that took quite a bit of study. It was very hard to try to get the connectomes to become what we needed for the functions that were being put in a naturalistic way if you try to do everything at once, if you just try to get the end result. After much running into walls and not seeing much progress, I realized that it was necessary to take a step back, take some perspective, and that's when it became obvious that you have to do this in 2 phases, sort of like in animal brains when you have these 2 different phases during neural development, where you've got the proliferation phase and then a pruning phase. So let's move on to that next slide then. So here's what we got out of that. This is the example of the layers, the architecture that was needed to be able to implement that XOR example in there. So on one end, there's a layer that is a set of pyramidal neurons that receive input. Then there's a layer that is full of interneurons. And then after that, another pyramidal layer, and then the output layer, which is also pyramidal neurons. And there are 2 axonal pathways from the input. One goes straight to the other pyramidal layer, and another one goes to the interneurons. And yeah, it not only did it take a little while to figure out that we had to do this in 2 phases, but we also had to learn that in order to get the axons to grow along the correct pathways and do multiple pathways from one neuron, for example, that we needed to have different attractor and repelling molecules on different growth cones of the same neuron, and that sort of detail. So there's actually a ton of study that went into just making this possible. And it does overproduce synapses. In this little example, it produces just 1,003 synapses. Those are multiple synapses from in each connection from a neuron to a neuron, and multiple receptor types on some of them. For example, the pyramidal neurons have both AMPA and NMDA receptors as well. So yeah, that was a bit of a balancing act, figuring out exactly how to get the proliferation to work correctly. And because of that, I think even just with this study, we've already got quite a bit here that we should write up and publish, just to get these underlying methods of the challenge out there as well. All right, next slide, please. So then we get to this next phase. The next phase is the tuning and pruning phase, which again resembles one of the phases you would find in human neural development. And what happens here is really that we're taking this connectome with all of that potential in it, all of those possible active synapses, all of those possible connections, but we want some of them to be very weak or to be eliminated entirely and others to be strengthened. So we're using the I/O function that we're looking for to entrain that, to tune and to prune. And for this we use, now that we've moved this model then from NetMorph, which is producing that original reservoir, we've moved it into our other platform called NES, the Neural Emulation System, which is our main system for doing virtual tissue development and virtual recording and all that stuff in it. We moved it into there, and then we're doing this pruning and tuning so that, from the original set of synapses there, we'll find that most of those get pruned away, and we end up just with a few connections. We made this quite minimal, so there are only very few neurons actually involved, almost as few as you could get away with, almost, but not quite, to create this XOR. Let's go to the next slide. And then, the next step is that we test. We want to see if that worked out. So we test if the function is actually there the way we wanted it, and we provide input to those neurons that are being used as the input neurons here and see what happens at the output. You can see, if you look carefully at the little timing differences there, you can actually see that the cells on the bottom are the input neurons and that it's propagating through to the ones in the middle layers, the interneurons and the middle pyramidal neurons, and then to the output. And you can see how, for example, the input to some of the interneurons there is inhibiting the activation of the pyramidal neurons in the middle layer. So we're getting the result that we want. We're getting this A XOR B being expressed as A and not B, or B and not A. And what you can also see is that although these layers, these regions, have a specific purpose, they do something kind of, it's sort of similar to what we see in most computational neuroscience models, where the different layers or regions in the brain are involved in a different way in the cognitive function or the neural circuit that we have. So you have that here as well, but the allocation of what neuron is involved exactly with what particular thing is, that's an outcome of both the outgrowth that happened in NetMorph and then the tuning. It's kind of coincidental. So we have two cells here that are involved in input B. We've got one cell that's involved with input A. Then we've got two interneurons that are cross-connected then to the pyramidal neurons in the middle layer, and so forth. So, but it's the neurons, the specific ones, is not something that we choose. It's, it just comes out of the function in a sense. Let's go on to the next slide. All right, so then there's step 4 and 5, which is really a step that isn't entirely complete at the moment. So some of this isn't shown because we're still finalizing the work on it. I'm putting the two together, 4 and 5, because the fourth one isn't quite complete. So this is the process where, as you saw, we generated something that's pretty sparse. We generated this neural circuit that does something and that has a somewhat realistic structure. Exactly how realistic depends on things like, do we actually express synaptic spines? Do we actually express postsynaptic densities in a certain way? All these details, which you can tweak, you can kind of turn the variables on and off in our model. That's for those details. But then on top of that, what does it actually look like when you start imaging in tissue like that? The systems we're working with, we assume that they are, neuroscience typically assumes that they are distributed, that they might be sparse or dense. So not all of the cells in this tissue are going to be involved with that function. And there's going to be a whole ton of other stuff normally. You'll find glia and vasculature and all kinds of other things. Now, do you want to deal with that or not? Well, if we're dealing with an early level in the challenge, let's say we don't want to make it too hard for someone, then we might choose not to add all of that. It could be hard enough just to retrieve the function from what we've got here. But In another stage, yeah, what you would do is you'd embed this in further tissue. You would hide this function in the remaining stuff that's there. And that's the part that we're not showing here. But yeah, the next part is that you then do the data acquisition. And what data we acquire depends a little bit on what the challenge taker needs, because some people work with electron microscopy, some people work with calcium imaging, some people work with something else. We're mostly focused on electron microscopy here, and I think we'll be adding expansion microscopy very soon, because that's kind of been the type of data recording that we think has the most promise for actual brain emulation, because of the huge quantity of data that you need and the high resolution, and you don't want to miss anything. So that's why we're focused on that. But yeah, so we then do virtual electrodes that we put in certain places and do electrode recording. We do virtual calcium imaging from a certain angle. You can do light sheet, for instance. When we do virtual electron microscopy, I'm not sure if that's the next slide coming up, but let's go to the next slide and see. I forgot what was going to be there. Oh, it is. Okay. Cool. Yeah, so yeah, as you can see, this is NeurogLancer. We use that. We spawn NeurogLancer from NES to then give someone an opportunity to take a quick peek at some of the data that's being generated. As you can tell, there are very specific variables that we've chosen to make this output. So some things are included, some things are not. There's a certain choice of focus and contrast. There is, you don't see any clear cell walls here and things inside of cells. So there are some choices that have been made on exactly what we rendered, but you generate these data stacks from which you could in principle then reconstruct in 3D and then start doing identification on them. That's the type of thing that ends up in these example data stacks that are then provided to a challenge taker. All right, let's go to the next slide. Now, when someone has or is taking the challenge, they use whatever method they want to try to analyze the data, to reconstruct, to discover the underlying function, in other words, the emulation. And the challenge takers are expected to return to us the functional neuronal circuit that they think they've discovered, along with the method that they've used. And we actually hope to use the latter, the method, to also independently apply to additional data as kind of a test of generalizability. That's still future work, but that will happen. Then we have another server in our system that isn't the NES server. It's not our neural emulation system, but it's the EVM. It's our evaluation system that is applying metrics that go along with the various success criteria for emulations that we're developing over time here. We only have a few right now. We focused on the ones that are relevant to neural circuitry and not to larger systems like cognitive success criteria and whatever. But yeah, so there's this system that then takes both the ground truth system that we use to generate the data and the proposed emulation and runs it side by side. We look at the structure, discover which pieces of the structure were correctly discovered and identified, what wasn't identified correctly. We generate a report about that. And similarly for the function, we test to see what happens when you put in random activity, when there's spontaneous activity, or what happens if you run the input-output, if you run the stimulus that is meant to produce the input-output behavior that the function we put in does, and see where the differences are. So you get another report about the function of that emulation as well. And together it produces a score so that we can compare the performance of different methods. Let's go to the next slide, please. Yeah, and this, just for fun, is the other system. So this is not the XOR, but this is auto-associative memory. Just a little fly through, just to show you something relaxing, because this might be a good moment to pause before we get into the next section. Are there any questions so far? or issues or discussion or anything really? Such a quiet crowd.
I was wondering, how you need to deliver the findings or the function actually.
I was having a little bit of difficulty understanding you there. Could you repeat yourself, Dirk?
I was wondering how you should deliver the function. How you would I heard him not say it actually.
Does
So,
okay,
so to have what you found, what function is, how you deliver to
okay. So I'm afraid I've tried to understand what you're saying by using closed captioning, but even then, it's telling me, so Jeff, where you found that food chain to. So I think it's not coming through well. Do you mind writing your question in the chat instead?
I'll do that, yeah.
And in the meantime, I'll take Miranda's question.
Yeah, so maybe you explained this, but if you could just explain again. So how do we know that your ground truth system that you've developed, so that's sort of like an artificial ground truth system. So how do we know that that actually strongly reflects the biological organism or the ground truth system, which is like the ultimate ground truth system, right? Is the organism. So how do we know that our ground truth system that we developed actually resembles?
That's a very good question. And I have a feeling Ken is going to jump in on that one as well. So we don't. That's not something that we can take right away. Really, what we're trying to do is we're trying to give people something that is beginning to approximate and increasingly approximate details of real neural tissue and the real data that you would obtain from it. And we want to see if these methods are able to handle what we consider a simpler case first, if they can handle what we're giving them. Now, there is a possibility that there's a divergence there, right? That you could say, okay, my method will work on real tissue and real data, but it can't possibly work with yours. Well, in that case, we've generated the wrong data. We're aiming to generate the type of data that can be used with those methods. So, for example, identifying cells by their structure, by their connectivity, by their axons and dendrites and what they look like, but also the connections they make with other cells, identifying the ion channels and their strengths by looking, for example, at the size of postsynaptic densities and how dark they are, that sort of thing that you would do in EM. Various details of how you would obtain the data and what you expect to see are the sort of thing that we put in here. But we don't expect the result to be something that is just like the real tissue. It's more like that case that you saw in the AI data set where, oh, can you recognize the schematic of a bicycle first, right? And then the next step is maybe, okay, now can you recognize the bicycle under snow? And so you're slowly getting towards something that is more and more realistic. So we're doing that while at the same time being very flexible and having many variables that we can turn on and off to create laboratory conditions for it. But you're right that this is never, ever going to be the actual test case. It's just that this is a case where we can train and where we know what you're trying to discover, so we can say something about the strength of the methods. So one thing that I another day when I was talking about this with someone, it came up as kind of, well, if you claim that your method just reconstructed some part of an animal, but we can't see the entire animal operating because it's only a little part, and you claim this works, you claim this is correct, great. There can be some evidence that you get straight from, say, activity recordings in the same animal. Perfect. That's nice. That's beautiful. As the system gets a little more complex and the neural circuit you're working with gets more complex, that can be a little harder. But if your method is able to find the function that I hid in here, and it's able to reconstruct that, and it does well on that, and then it also does well on, say, neurons in culture, if that's our next step, and it does well when you're reconstructing and you're using, say, co-registered activity data as well. If you can do all three of those, then I have a fairly high confidence in that method compared to one that can't do any of them or can do only one of them or only works in one case. This allows us to run the method through a lot of different examples and to test it in that way, to test its generalizability more. So, and I'm not saying it's, like the magic bullet or anything. It's meant to address a specific problem, the problem of having to work in systems where you don't really know if you're discovering what you're trying to get out of it. And there is going to be a slide that's a bit more on that, basically on the question of what are you trying to find when you're doing translation. Yeah, we can get into that later. Okay, so let me just quickly grab Dirk's question here before I go to Ken and then Richard. So Dirk's question was, my question is what we need to deliver. We need to find the function of the scanned data. Do we need to deliver a simulation or some function description we're supposed to find? Right. We are actually still working on finalizing exactly how the challenge protocol is going to work. I think that what we're going to require is to deliver the simulation so that we know what you found, the model that you derived, and the way that it's implemented and how it works, so that there's no there's no misunderstanding, no doubt about that. So we need to have the simulation, I think. Ken, yes, you have a point or a question.
Yeah, Randall. Thanks. This is it's a very good explanation of everything. So I'm sure you've thought about this. So let me try to articulate it. So the challenge is essentially each individual challenge is made up of different component parts. If I think about it, so one of the things that a team would have to do is they would have to look at the fake EM data and do segmentation on that. That is an algorithm that can be, infinitely developed. Another thing is to look at the electrophysiology data and extract essentially that this is doing an exclusive OR. That is a spike counting type of thing. For the memory, auto-associative memory, if you had the connectome, going from the connectome to a set of attractor states, that is a computational problem that goes from connectome to this, and each one of these is a different algorithm that that any particular group might have great expertise in. But you're kind of putting out a challenge there that is going to, I think, repel some of those groups that are very specialized in certain things but have no expertise in other things. And I'm wondering if there's a way to divide up these challenges so that individual groups that are, let's say, very good at going from a connectome, a connection matrix, to function, are not forced to develop a segmentation algorithm from scratch, and vice versa.
Yeah, that's actually a fantastic question that has indeed, of course, come up. And that, so this is still a work in progress, and this is an update basically on where we are. And just the full honest truth is that we have not yet figured out entirely what our challenge protocol is going to be. Whether we need to have, like, how many different groups do we need to have? How many different levels with different types of parts of the challenge, like you said? And for example, does every challenge taker need to solve, say, one problem that is unique to them so that they cannot in any way have learned about what's in there from the results someone else published, and also carry out the same thing using their methods on some problem that is common so that you can compare between different challenge takers better? So I agree that that is a big problem. And getting people to want to take the challenge is a big problem, partly because of what you said, that it might turn some of them off if they feel like what they're doing isn't best served by trying it on this challenge, and for many other reasons. So that was one of the first things Conrad Cording said about the challenge. Was he said I think that we definitely need to have ground truth systems. It's very important, but it's going to be extremely hard to get anyone to take the challenge. So yeah, that's definitely a problem. So let's just say I understand what you're saying. I recognize the problem. We don't yet have a worked out solution. All we have is an understanding that that is a problem, and we need to figure out a protocol that really works for that.
I mean,
Actually, one of
I
the
think
peer participants here had come up with some good ideas of how to set up the challenge, and we have to pick that up with them again in a little while. Sorry, yes?
I was going to say, I mean, I think there is definitely an advantage to having an end-to-end challenge as you presented it, to kind of make people understand that these sub-challenges are part of a larger program. If there's some way to not lose that big picture aspect of it, but also provide a challenge to particular groups that have particular expertise, like people that are really good at segmenting neural circuits. I think it can be a win-win type of situation if you figure it out.
Yeah, actually, I'd love your input on it as well. So if you feel like thinking about the problem a bit and maybe coming up with some ideas there, that'd be fantastic. Because yeah, we really have to work out the protocol. Richard.
Hello. I don't know if this is a good question or not, but there was a slide with like those black and white images, right? The microscopy images. Are we able to produce those realistic microscopy images from the fake EM data stack?
You mean, are we able to produce, say, the images that you saw from Genelia?
Yes, I'm not sure if I'm familiar with.
Yeah, there's still some work we have to do there. For example, we don't have any components besides cell bodies, axons, dendrites, synaptic connections and their spines and things like that. We don't have, say, any glia right now. We don't have vasculature in there yet. And also some of the details of how we generate these neurites. While there are some things that we can tune right now, there are still some things missing that we really should be able to do to give it more of the appearance of what happens in microscopy. And that includes also some of the artifacts. So we have some artifacts in there already, some kinds of interference that you can see, some kinds of noise and so forth. But again, it's not complete. As you can imagine, there's a lot to do here, which is why the last section of this is about what do we need, because we really need help to make this thing bigger than what our tiny little group has been able to do. So yeah, there's still some work to do there. Thank you.
Awesome.
Did someone just raise their hand again? I'm not sure if... oh, Derek wrote something. Yeah, it's nice to use this event to form teams. For example, I'm a software developer and writer, but I need more knowledge about the biology neuron part. Yeah, excellent. Yes. I would love to hear towards the end more about anyone who's interested in helping out in one way or another. Absolutely. And yes, programmers very welcome. Okay, so I think actually, since we just did kind of a break here and we talked a bit, I would love it if we could take, say, maybe a 3-minute break or so and then come back 3 to 5 minutes. Is that okay, Thomas? We can do that?
Yep, sounds good to me.
Awesome. Okay, so see you all in 5 minutes. All right, refilled my coffee cup, put on my shades. Now I can't see anything.
All right, I'm back as well.
Yeah, probably to give it another minute or so. And then we'll dive into the remaining parts.
Sounds good.
I think the remaining stuff isn't even as long as what we've done so far.
Yep. We also just might want to mention, maybe we could direct people to the BrainGenix website so they could see a bit more information on the software and all that.
Yeah, do you want to type it into the chat or something?
I can. And also, I don't know if our Git repositories are in here at all, but you could always link people to those as well.
Yeah, I didn't show them or mention them.
Yeah, no worries.
We can point them to it.
Yeah, sounds good. I'll put links in chat in a minute.
All right, so I'm guessing that most of you have returned at this point. Thomas, to get this going, let's go to the next slide so we can see the next topic coming up. All right, so this is more of a, okay, so now you see what the challenge is and why we're doing it or working on it. And so then we're kind of coming back to the question of the problems that are actually, that we want to see solved, that we want somebody to solve, that we'd like to help solve. And last time I mentioned only the translation problem, but there's also the scale separation. Actually, I did mention the scale separation briefly. Yeah, these two problems. I think that the brain emulation challenge can help with both, or it can become something that will help on both. Let's go to the next slide. All right, so I'm going to run through, in the next 3 slides, I'm going to run through, say, these subtopics and maybe make a case for that. So when you're working on brain emulation, it's important to know your aims. So what you're aiming for, what you're trying to achieve, and to validate your methods such that you can achieve those. I mentioned this a little bit going into it. And doing so, knowing what your success criteria actually are and what you're aiming for, that's going to prevent you optimizing for the wrong thing. And I'm going to say something that's perhaps a bit controversial here. In that third box, you can see already in red. So these success criteria, I'm claiming, or we have a paper draft that's about this, are really about our cognitive capabilities, about our memories, about things that are unique to a specific animal or person, such as the memories that are unique to a specific animal or person, but that are about that functional content rather than about the specific way they're implemented or the way that that content is implemented. So I would say that validated emulation really isn't about specifically about the 3D fidelity of your data stack. It's not about the biological components that you have, how many you count in there, whether you've captured every mitochondria and everything in there. It's not about ion channels necessarily either. So the true test then is this system function expressed as the sum of all of the circuit functions. And yes, there is an underlying assumption here. This is really based on this functionalist approach to brain, mind, and all of that. So solving the translation problem has to tie into this verification and validation thing. This is the obvious part of how the emulation challenge fits in, because this challenge focuses specifically on giving a score that is based on this idea of the success criteria, and using these known ground truth systems for that. Can we go to the next slide? And then again, still about the translation problem, some of the things that this gives us, it gives us kind of a laboratory where we have absolute control over what's there. It's super flexible, the system, where we can try to match the problems that our challenge generates and the data that it generates to the types of methods that people are trying to use. This ties in with the comment that Ken made about people doing different things and having different methods that are aimed at, say, one way of acquiring data, say whether you're doing electrode recordings or you're doing EM. So you'll want a different type of data to work with that. The challenge is flexible enough to do that because it's virtual. So we can tune this to electron microscopy, we can tune it to expansion microscopy with molecular annotation or calcium imaging, voltage imaging, and we can decide to tweak other variables to make the challenge harder or easier depending on where we are at that time, what type of work people are doing, how well their methods and algorithms are working. So we can tweak how sparse or dense things are, and whether specific things like the postsynaptic densities are easier or harder to identify in the data, and anything really. And it begins to approach along these different tracks for different types of data, different kinds of methods, begins to approach eventually something that becomes not quite realistic, but realistic enough that you can then say, okay, I've tested my method and I have some really rigorous results where I can say, here's why I think my method is really good at discovering X. And now I'm applying it to this data that we don't really understand, but where we have some theories and some hypotheses and some co-registered activity data, perhaps. And maybe there are systems where you don't have that co-registered activity data, and you can apply it there. So there's this transition that it makes possible. So this virtual brain platform that's in there, that explicitly supports hypothesis testing and the honing of these techniques, these two things. So that's how, this isn't surprising, because this whole way through we've been talking about the translation problem, and that's the reason for this challenge. Now, the next slide, please. So I think, even though that wasn't the original reason why this project was developed, I believe that we can also help study the problem of scale separation. So this question of at what resolution do you need to emulate what's going on in your biology to get the result you're looking for? So for these success criteria that we have, the thing that we're trying to get out of it, the kind of brain emulation we want, or the kind of neuroprosthetic we want, what do you need to identify? So what data do you need to acquire? What do you need to model, and what needs to be correct there for that to work? And when do you lose something? When do you not get the function you're looking for? When do you not get, say, let's just jump right there, consciousness? Sorry about the dramatic effect. Anyway, the question is, in the scale separation thing, it's partly just about, okay, what resolution do you need to get to? That's the assumption that there is a scale where you can separate these things. There's a bigger question even where, there are some claims, let's say Miguel Nicolelis, who believes that there is no ability to do scale separation as long as you're working with digital systems, because the analog will take you all the way down. So there's even the question, can you separate scales? And being able to separate the scales to find a highest resolution that you need to study carefully and that you need to recreate, that there is such a resolution where you can do that, and then you can move it to a different platform, a system like a computer that can carry that out. Well, that depends on not just the functionalist assumption, but also this idea that there is a place where you can do scale separation, that you don't have a chaotic system where you cannot separate those scales anywhere. But here, I think that we can at least start comparing and studying a few things, even if we can't solve the whole problem, because of course these are generated data sets. It's not biological data. We can at least compare some claims, like we can compare claims about, is there a lo-fi way to do whole brain emulation that will still give you the cognitive functions you're actually looking for, that kind of thing? Can it do those things? Can you get that back? I think there are some things that we can explore in there. So maybe not the whole question of scale separation, but some of these, by just directly comparing and testing different forms of data acquisition, low-fi, high-fi, and different forms of reconstruction emulation, again, kind of in those categories. So for those people who are interested in these topics of, can you do lo-fi whole brain emulation, or do you have to do hi-fi whole brain emulation, what we might call classical whole brain emulation, this could be an interesting platform on which to directly test some of these ideas or claims, even if it is in a virtual setting, because at least you can run those ideas through something concrete first, and you can then make some statements after that. This is actually something I'm not sure if it's really applicable there, so if you guys have some ideas or thoughts about it, please speak up. I'm trying to handle in my mind this whole concept of the lo-fi whole brain emulation approach, since it's become kind of a popular topic in some circles. And yeah, so that's led to this. Yes, Richard.
Could you describe more, like, I guess because there seems to be like a split in the neuroscience community between lo-fi and hi-fi. Like, could you describe that split more, I guess?
Which split are you referring to in the neuroscience community? I know that there is an interest
Bet-
in some sort
Between
of.
lo-fi and hi-fi, like, methods of building stuff.
Okay, so in the neuroscience community, I'm not really aware of that so much. I mean, obviously, there are many models that operate at a very abstract level. And there are also, of course, many data acquisition techniques that are very lo-fi, like MRI or EEG. So those things definitely do exist in both high fidelity and low fidelity, or high resolution and low resolution. I'm not sure if there's a large discussion in the neuroscience community about trying to, say, recreate something that resembles an animal brain or a human brain at a low fidelity and getting the results out. But I do know that there's interest coming from communities that are sometimes neuroscientists, but that are also sort of involved with AI and questions around that. I've been having a hard time wrapping my mind around exactly where that falls and what kind of theories or ideas these are. I wouldn't necessarily call it a theory, but claims or hypotheses. I'm still trying to understand that better. But I mean, if people feel that it is an important enough topic because maybe it can be developed faster or something like that, that they're willing to put a lot of energy and a lot of resources into it, then I think it's worth testing these claims as quickly as possible to try to see where it can actually go, and how much is that even whole brain emulation, or is that something totally different that you're talking about if you're taking the fidelity down that many notches. I know that some of you here have dealt with that topic a little bit. You've at least talked to people who are looking into this idea of lo-fi whole brain emulation. So maybe you have a better understanding than I do of what that means. But I felt that it's important to be able to test this quickly, especially if there's a question of what sort of projects should one pursue. Are there any more thoughts or ideas about this before we move on?
Thank you.
Okay, let's go to the next slide. All right, this is not going to be too long because you've already seen quite a lot of this, but here's just kind of an overview of our state of work. Let's go to the next slide. So we've put in somewhere between 12 and 15 months of work on the platform now, and there are only a few of us who've been able to do this full time. So it's a pretty small team, very tight knit. We've been working hard on this issue. But yeah, so we came up with a construction language for these three-dimensional dynamic neural networks in that NES system. We built the back end and the visualizers and all sorts of details around this. We ended up integrating it, of course, with NetMorph to be able to do the sort of the outgrowth phase, that proliferation phase where we build the reservoirs, the connectome reservoirs. We integrated it with NeuroGlancer as well. And as you saw, it's gotten to the point where we can do most parts of the challenge right up to data generation, which still needs some fine tuning and tweaking. But yeah, actually, we should go to the next slide to see what's still coming up there. Right, so we still have some development steps left before we can really launch this project. And I'm wondering if it clearly says on here that we need to figure out the protocol, because that really is part of it. Maybe I wasn't thinking of that as development since it's not technical in that sense. But okay, yeah, we need to improve the efficiency of our simulation system, even though it is parallel and multiprocessor and all that. It's getting to the point where running the development of larger systems and then also rendering these data sets at scale as you get them at larger data sets, it's hard. It takes a lot of computation. And so the more detail we want to stick in there, the more efficient that has to be. Right now, we can say that at least generating that data is still running a little bit faster than acquiring it on a high throughput microscopy system, electron microscopy system, but that's, barely. We need to learn how to embed more components. So we need definitions and ways of developing more of these different components like glia and vasculature. There's still some fine-tuning work that needs to be done on our data generation and the different variables that you can use to tune up and tune down the difficulty levels. And I guess that gets into the protocol a little bit, as well as, of course, the type of data. That's part of the protocol question. Then, really importantly, we need to provide some example data sets that aren't the type that are being generated for someone to take the challenge, but that instead are data sets and examples where someone who's considering taking the challenge can see what that looks like, what that is, what they would be working with, and also how it gets evaluated. So we need to prepare those and make those available. And that also will give us an opportunity to get a lot of feedback from the community about them and improve them further. We need to prepare a bunch of these examples for generating multiple cognitive circuit types, these buckets that we were talking about. And there's a bunch of publication work left to be done, partly about improvements that we made on the NetMorph system and about the platform itself, but also about the verification and validation process. So about the success criteria, the metrics, and how they're applied in this challenge. So these are still quite a few bits of work. Let's go to the next slide. Okay, so this next bit is kind of about what we hope is going to be possible with the challenge. So why we're doing the project, what we want to get out of it, and sort of how we see a timeline going, assuming that we get enough resources, enough help to make it happen. So let's go to the next slide. So the first thing, let's just call it the launch here. This would be, at the beginning, a soft launch for some of the reasons that Ken already alluded to, that Conrad Cording pointed out, that other people have been mentioning, which is especially when the system is still young, when it hasn't been demonstrated, when there isn't a reason to believe that it has the value that someone wants, or maybe you think that your methods aren't really testable with that system, or how do you compare it? Nobody's seen it. Really hard to get challenge takers. So the best thing to do is to launch it by publishing what it's about, publishing how it works, and by yourself and with your partners, that a few partner labs, Conrad Cording's lab is one example, taking that challenge, applying your best methods to it, maybe going through the literature and finding examples of reconstruction approaches that attempt to functionalize that we can then take to the challenge, publish something about it. It's there's also a slight psychological aspect to that. Sort of, okay, if so-and-so has published something and then we go along and we use what they've published and we try to apply it to a data set and then publish our results with it, then there's a little bit of this sort of motivation or push where said author might say, oh, you did it wrong, or I could have done it much better, or the method that you used to test it just isn't right. And then, you start a conversation, you get some feedback, get some visibility, maybe they try it. So this is a tough phase, the launch year. It's a difficult one, but we have to get through it one way or another. And there will be an aspect of, I guess, communication and marketing about it as well. But then hopefully, if we can pull together the funding needed for it, there will be a first prize challenge. Once we know exactly what the protocol is for this challenge, and maybe there will be several categories, we'll see. And, these results will deliver at least some form of transparency. Maybe in the beginning, people won't agree that this is saying about a method what they wish it would say, but it will at least produce some results that you can compare. And then it's up to the rest of the world to either come up with a better challenge, better ways of competing and comparing, or to participate and show what they can do. I guess this is all really part of how do you get a challenge to even work. And it's not unique to this challenge. It's something you have to do to get challenges to work. But then hopefully by the end of that year, you've got some interest raised in both academia and outside of it, because one of the cool things about these AI data sets and challenges, of course, is that you've got a lot of people participating from say startups and other places that were not even involved in machine learning research previously, and that then got interested. So the amount of research hopefully would start multiplying. That would really be, even if we just start to get that, that would already be solving one huge problem in work on translation right now. So that would be great. Next slide, please. All right, so then once it's actually launched, hopefully there would be ongoing prize challenges. And we're imagining something like a 5-year stretch where you get increased competition. There, of course, as a result, there are additional demands where they'll say, oh, no, we need this kind of data, or you're not doing it right, or this isn't realistic, and so forth. There's going to be, I guess, a little controversy. And that will require more development on our end. So the team has to continue developing this as the competition heats up. Then there would be a first batch of, say, verified and validated methods for which there is some rigorous description of what they work well on, what they don't work well on, and why. There might be some high-profile challenges, and as you see in the box down there, so multiple tiers of the challenges, and you'll start to see. So Ken mentioned specialization already, but I'm imagining that once you have these challenges, you'll get even more specialization, that there are different methods that people just keep on improving. It's sort of like in AI, where some people then bet on deep learning and other people didn't. You find different ways of addressing, say, computer vision. And then you get scientific publications based on those results. And hopefully at that point, we've got a feedback loop that's beginning to ramp up an iterative improvement. So in that case, we would have the start of that loop that I showed in the beginning. This would be so over multiple years is the assumption, but yeah, resources make a big difference here. Next slide, please. And then comes the part where hopefully, because more people are doing things and because there's all this very rigorous and concrete comparison going on, that you start to get very clear differences in performance for some classes of methods for some types of problems, and you get meaningful insights and you get some paradigm shifts going on. So then what you get is specialization in labs. You get more journals that talk about this. You might even get, a translation journal that isn't about language, but is about brains. And you could get, and that was a joke, by the way, you get technology development and more investment in it. So this is this sort of positive feedback loop, where what we would love to see, of course, is going all the way back to kind of closing the loop with the beginning of this whole discussion, this whole talk, is we'd love to see this proof of concept, to see results on biological systems that are rigorous, verifiable, and not trivial. So not a biological system that is trivial, and then scale up in performance and capacity. So then you get, a solution to the translation problem, and that helps out brain emulations. We're almost at the end here. Thomas, can we get to the next slide? Thank you. So this last section is what we need to help us all succeed. And I know you guys aren't all investors or donors, but we need to get the word out. So let's go to the next slide. We have been trying hard and will continue to try hard to find financial support that'll make it possible for us to continue working full-time on the development of this channel, and even to increase the rate at which it gets to its goal, and maybe make that first launch really successful, and all the other stages as well. And kind of analogous to the stages that I mentioned before. So here we see kind of a setup phase where we still need to complete a few things. We need to have enough staff that we can actually run the challenge and that we can generate data that we need, do publications, and so forth. And you see here that setup phase, we assume, will come in at something like 340K over the next 7 months. And then it's time to launch, the actual launch. So we have some people who've been gracious and want to be the first challenge takers for us while we're still testing this. And that is wonderful. But there comes a point where we're really launching it and making this an open challenge to everyone. At that point, we of course need to continue working, because as mentioned, people will have all kinds of demands and there will be a need to keep on tuning the system and to react to whatever is happening there. Plus, of course, just running the challenge itself is something that that takes people, servers, and so forth. So this has a cost attached to it. Again, there's something that is, the price tag there is something we've calculated over what that would normally take. And similarly for further iteration of the challenge. And there's the possibility that the prizes are in there, and those prizes could be, hopefully, if the prizes are larger, if there are more, say, different prize categories that we can have, then that will again accelerate things. So there's a lot in here about how you allocate resources, what you can find as resources, or if it becomes more popular and we get more investment in it, more donations, more resources, more sponsors, more support, that it can ramp up both in breadth in terms of what we can handle, and in depth and in speed of progress, and in getting people to take the challenge and work with it. It has been great to work on this. It's wonderful to be working on something that I feel is very clearly and directly relevant to moving the needle on getting to brain emulation, whole brain emulation. It has been, as it has always been so far, fairly challenging to find the necessary financial support. And I would love it if anyone here can help us to make the right connections and to find the resources needed for this. It's not a very big project compared with a lot of other proposals that are out there. This is actually pretty carefully curated. It's something that has high leverage potential, that will bring in people from academia and outside, that will increase the number of people working in this field, that will increase attention, visibility, motivation to do it. And for a relatively low price tag, when you compare that to, say, for example, the whole mouse brain project, just to mention one, right? We're talking about a fairly low price tag, but we need to find the right connections to the right interested individuals or groups or organizations that actually care about moving the needle on this and that understand what we're trying to do here, so that we can really move it ahead. So I encourage you to study this slide, to either critique it or to work with us on it. But really, we need some help at this point, because we've done months of technical development, and we've tried our hand at reaching out to the right groups that would be interested in supporting what we do here. But we have yet to get to the point where we have enough funding to even continue working on it full-time ourselves. So this is really crucial at this moment. I can't overstate how important that is at this time. And with that, let's, I see some notes here about Y Combinator, and that's interesting. Okay, let's get into some conversation then. I think there's might be one more slide, but it's just the thank you slide, I think. Yes, it is. Yeah. I just want to, of course, express our gratitude and thanks to those supporters that we have. We do have some supporters who've helped us be able to keep our servers running, to work full-time on what we're doing here. And you can, of course, help us directly as well. It doesn't have to be you don't have to be, a millionaire putting in lots of money, but even any donation helps, really. Even just keeping our servers going, being able to put in $50 here or there for more disk space or more RAM or whatever, it's all quite important. So if you can, please do. Thank you so much for being here and for talking with me. Let's continue the conversation until we run out of things to talk about, or until, I don't know, we get tired. So, floor is open.
Randall, do you want me to stop presenting the slides, or?
Unless somebody wants to go back to a slide, I guess. Yeah, let's stop the slides for the moment anyway. Yeah. Or you can keep this one up if you like, because then at least, in the live stream, they'll still see that link. Yes, Yunsuk.
Right. Thank you for the presentation and getting this started. So I had some ideas about validation. So how do you validate emulated model against the ground truth? So one is, I guess, a real interesting one is to lesion the ground truth model and measure the outcome, and do the same in the emulated model and see if the lesion model works in the same way. I guess this will allow you to avoid any kind of overfitting in the emulation. And another idea is to give a new, novel task. Instead of the original task that you published, some kind of cognitive task, you can set up a related task that has not been published through the contestants, and then again see whether the emulation exactly replicates the expected result if you run that new task on the ground truth.
Thank you. Yeah, those are some great additional ideas because, yeah, the only kinds of validation that we had been thinking about besides the structural one so far was testing with the intended data or stimuli for the function that we're looking for, or basically a sensitivity analysis, some kind of random spontaneous activity. But what you're saying, the lesion thing, that's a wonderful idea. The other one, giving it a different task, I think in the large examples where there is a lot that circuit can do besides this function, that will probably work. The XOR is not a good example for this. It's too simple. But yeah, I think that in the other more complex systems, more involved systems, that you could do that too. You could see what other, that's sort of like the regular thing you would do if you're carrying out a manipulation, doing some stimuli and then seeing what happens if you're then, recording from your neurons. So we can do the same thing in the ground truth and in the emulation. Yeah, both ideas are great. Yep, definitely taking note. Very glad this is all being recorded. Yep. I just noticed here that Ocean said could possibly contact Y Combinator as well. I'm not sure if you have any further knowledge or connections about that. I'm wondering if, to your knowledge, is Y Combinator interested in this sort of thing? Because right now, this is not I don't see how this would be, say, for example, a for-profit startup. Like, what kind of product could come out of it? So it's more of a non-profit thing. I'm not sure if Y Combinator does that. Okay. Yep. Yunsuk?
Yeah, so I have another comment. So when you develop your ground truth model, to generate the dynamic activation patterns, you might have to do some kind of numerical integration, right?
Yes. Yep.
Right. So in that case, the integration time step really matters. So it would be, I guess, very important to actually make sure that your ground truth model itself is kind of robust. So
Yep.
slight changes in the integration time step.
This is true. And that reminds me that there is an entire subtopic that we didn't raise in this talk and that maybe will be in the next micro workshop or whatever. There is the whole question of constraints, because these tiny differences in, say, time step effects on numerical integration, those are very important when you're dealing with an analog system that could do a random walk in any direction. If you have many constraints imposed on the system, such as recurrent inhibition and pattern separation and all that sort of thing, then you can do away with some of these concerns. So then you can find a minimum time step, for example, that's safe. And I'm sure that's what you meant anyway, is finding a safe one. But there's a whole topic around constraints. So sort of like I had these two sections on, say, translation and scale separation, I feel like constraints is its whole own topic that's also really interesting and explorable in this case. Yep. Yes, Ken.
Randall, I was hoping that you might talk a little bit more about the auto-associative memory challenge example. What is the function that is supposed to be extracted from that in particular?
So I'll be honest that the auto-associative memory thing hasn't gotten past the developing of say the reservoir of connections so far. We haven't worked it out fully. But the idea there is, so there are multiple versions of it. There's the version where you have, say, auto-associative memory and heteroassociative memory. You're trying to do something that is, say, doing episodic memory or whatever. Just those interesting examples that are, again, trying to mimic something you would find in the neuroscience literature. But the basic example is just the ability to encode certain memories as attractors and to be able to find those memories, to find the representations that were encoded in the data, to be able to then build a system where you can either point to them and say, I see that these attractors are here, or you have a retrieval mechanism and you can demonstrate that those attractors are there. Interesting that you're asking that, because this sort of relates to your prize challenge, doesn't it?
I mean, it does to a certain extent, although the aspirational neuroscience prize is probably even less structured. But one of the reasons why I'm asking is I have really been quite curious about what the state of the art is in terms of algorithms that can look at a connectome and extract attractor states, let's say, or heteroassociative associations, or whatever you would call them. And one of the things that I would hope that I would get going to the challenge, the Brain Emulation Challenge website, let's say, would be a bunch of pointers to what the current best analysis algorithms are in these different areas.
Agreed.
So just to phrase that in the form of a question, how good are we at reading a connectome and getting attractor states?
Right. Yep. That sounds absolutely correct. Part of the task in making the publications we intend to put out there is to also include a really good section on background literature on what is the state of the art right now in different areas of analysis and reconstruction. And that would include something like that. Now, you're right that maybe that's better to put that just on the website and have it there as something people can browse through as they're going into the challenge. So maybe that shouldn't wait for a publication. Yeah, that's a really good point. Yeah. Oh, okay. Y Combinator does fund nonprofits. All right, that's interesting. That's actually super interesting. Yeah. Thank you. All right, let's see here. So we've been doing this for about an hour and 45 minutes now, which is maybe 15 minutes longer than I thought we'd be going. I do think there's still an opportunity for more discussion or questions if you have them, or you can reach me afterwards. I know that some of you expressed interest in either participating, joining our team, working with us, helping us, or you have ideas about how the protocol should work or what kinds of problems we're going to bump into. Now that you've heard a lot more about this challenge than maybe you've heard from me before, if you heard anything from me before, I bet you're going to have thoughts, ideas, critiques, and so forth. And we'd love to hear all of those, both about the challenge itself, the techniques or technologies, how it would be received and what we might prepare ourselves for. And of course, the question of how to even get this thing launched. So if you have any previous experience with getting a challenge launched or getting a project launched that is not coming directly from a university or, from like a simple nonprofit like us, and you've found out how to connect with the right people, or you have some ideas about all of those problems, I'd love to hear about them. So I think there are a whole ton of things we could do moving forward from here. And I do want to keep this conversation going. So we also really our intention is really to have more of these, and you'll see kind of progression, advancement as we go, but also slightly different topics. The next micro workshop might be as soon as 2 weeks from now, although I won't promise that. And yeah, in the meantime, I'd love to be in touch with all of you. So this is kind of the last call. If there are any more questions or comments at this time, this is the time for it. And otherwise I will, yeah, Thomas just pointed that out. You can reach us at contact@carboncopies.org. And some of you already know our direct email addresses, etc. anyway, so you know how to reach me. Well, thank you. This was delightful, and I hope it will also lead to good things. Have a great day. See you all later.
Yeah, thanks, Randall. Bye, everyone.
Bye-bye.
Thank you.
Thanks very much. It was really great.
Bye, Dirk.
Thanks, Randall.